This is one of the most beautiful and spectacular places that we've been, and so I hope this is the beginning of the real visit and a series of future ones. I really want to also thank Felice for using Wiley as an inspiration, who was really one of the defining scientists of our generation, and his loss was both unexpected and really a major shift because he spanned so many different types of areas from physiology and endocrinology and neuroendocrinology and peptide biology. And I won't say that he was an omnivore. He really was very focused, but he understood the power of peptide biology and didn't shirk from any part, from the actual setting up of in vitro assays, the complex purification, no matter how many hundreds of thousands of sheep hypothalami it took. I saw him load these giant columns and get little tiny fractions out of them. And these were important problems. The conquering of the Mount Everest of neuroendocrinology, which is the isolation and determining of the structure of CRF, was just a gigantic problem that had been trying to be solved for at least 3 or 4 decades. And with focus and brilliance, he was able to do that. And so it was a landmark event, and he deserves more than the credit. Is just the way in which he accomplished things was very impressive. And he always had a sense of humor and self-deprecation, and Wiley was a great pleasure to be with. I'm going to go to the slides. So the two stories I'm going to talk about relate to 2 nuclear hormone receptors called PPAR and FXR, fatty acid receptor and a bile acid receptor, in coping with nutrient stress and inflammation. And the underlying context is the Hunger Game. Now, Bill Crawley mentioned that reproduction is the essential evolutionary drive for fitness. And I won't tangle with him on any issue, but I look at it slightly differently, which is the ability to acquire nutrients efficiently from the environment is the essential selective force. You have to, for any organism, you have to be very good at acquiring nutrients in a competition with other organisms for nutrients. And so nutrient metabolism underpins reproductive fitness. That is, if you don't get nutrients, you will not reproduce, and we know that. So we have to think of the intersection of how nutrients come into our bodies and how we manage them. I'll tell you a little bit about nutrient stress and the inflammatory process. Now, some of you know Wiley was called W, or W, like another famous person from Texas. But he wasn't George W. He was just W. And there he is toting his 2 guns. We traveled with Wiley. This is up in the Andes, and we were buying serapes. And because of an error, we wanted to get alpaca-based serapes, and Inder Verma here got a really good deal from it. And then when we got them, the woman who we were negotiating with said, it's acrylico. And so we got a good deal going to the top of the Andes to buy acrylic serapes. But they lasted a really long time. And then here we are playing tennis, which we did weekly for about 30 years with Steve Heinemann, Inder Verma, myself, and Wiley in our early days. Now, just historical background on the physiologic mechanism. Claude Bernard is one of the towering figures. Back in about 1875, he began to illuminate in his book, The Principles of Physiology, on the mechanisms of the physiologic mechanism, where there are events and processes in the body that counterbalance each other. And if something goes up, there's a mechanism to bring it back down. If it's too low, there's a mechanism to bring it back up. He studied nutrient metabolism in particular. He was interested in the pancreas and the liver. And he came up with many of the ideas of the nature of the homeostatic mechanism and really was one of the key features in setting the groundwork for the future of physiology and ultimately molecular physiology. Now, I work on nuclear hormone receptors. This just shows you that in our bodies there are 48 of these genetic switches. They act in the nucleus to control transcriptional networks. And these are profound regulators, again, of three things that have been mentioned in this meeting. They're important regulators of metabolism, reproduction, and inflammation. Now, you can call everything physiology, but the nature of the way in which receptors do this is, I think, important to look at the transcriptional underpinnings, that nature chose a transcriptional mechanism to mediate estrogen and androgen and progestin activity, to mediate gluconeogenesis through glucocorticoids and control of sugar and salt metabolism. Yet, as potent as these physiologic regulators are, when you activate a metabolic pathway, you will trigger an inflammatory response. We've been hearing about that in the presentations. And so potent mobilizers of energy, and anything that puts nutrients into your blood will be a pro-inflammatory process. And so glucocorticoids can put in a lot of sugar. But at the same time, glucocorticoids, medically speaking, are almost used only as anti-inflammatories. And so nature builds into a single molecule, a single nuclear receptor, the ability to mobilize sugar as well as to suppress inflammation. And this is a very interesting, important thing to consider. And many nuclear receptor ligands are anti-inflammatory and pro-metabolic. And so we have to think about mechanistically the logic that nature uses to couple reproduction, energy homeostasis, and inflammation, because they're all intimately linked, structurally linked, I think, in signaling systems. I'm going to tell you a story that relates to two receptors that are called FXR. We isolated that in 1985, so quite some time ago, and the PPARs. FXR is a bile acid receptor, and the PPARs are fatty acid receptors, and I'll tell you just a story about one of them that relates to a new mechanism for insulin sensitization. Now, looking at how nutrients come into the body, we have to get the clock set here because I'm reeling along and it's the 20-minute clock, so yeah. No, I know, I'm just looking at it. I'm almost in the red zone. So one way to visualize nutrients and the importance of the idea I'm trying to get at is that nutrients just don't come in the body. There's a flux, there's a vector, there's a nutrient pressure gradient that as you eat, you now have calories that have increased in your stomach and your digestive system, which begins over here. Those nutrients in that pressure gradient then has to be transferred from this anatomic location into the body and often basically hits the liver first. That creates an additional internal pressure gradient for nutrients that the liver now is managing, and it distributes calories through circulation to the adipose depot. And the adipose depot stores in this pressure gradient if you do not use the calories for long-term use. But obviously the adipose depot is a very dynamic tissue, both structurally and functionally, and it provides, along with the liver, the principal source of nutrients to muscle that's going to burn most of the sugar and lipids in the body. So we have to think of how these vectors are actually controlled to manage nutrients. And nuclear receptors play a critical role in managing this. And also there is, sitting at the top, the brain. And we don't really fully understand how to integrate the brain into this, and that's what a lot of the people at the conference are talking about. But it's an important feature to consider not just to study one single tissue, but the physiologic system and the physiologic mechanism is distributed. And different tissues play different roles. And so because we're multicellular, some tissues are altruistic. the adipose depot is not there to burn energy. It's there to store energy for other tissues who will burn the energy. And so other tissues make the energy to keep this whole process going. So mechanistically, it's very important to, in terms of disease, to understand how to control these vectors, and I'll tell you a little bit about that. With weight gain, you saw some of the studies and the, oh, sorry, I'm going to go back. With weight gain and aging, so when you're young, you tend to be much healthier because you're physically active, your body's in a great state typically. But with high-fat diet and reduced exercise as we get older, and our genetics, we can progress towards obesity and metabolic syndrome or syndrome X. And now this bathing suit and pattern that's on this young guy is now on the chair he's sitting in, and now he's a couch potato. Instead of being a surfer, he's a channel surfer. And that's what happens. We get locked into being sedentary and overweight. This leads to insulin resistance, which is the root cause of many problems that lead to diabetes, hypertension, hyperlipidemia, heart disease, and cancer. And so you do not want to have high insulin and high sugar in your blood. Both are actually pro-inflammatory individually. And so you don't want to have those two elevated because you're going to have a problem. So insulin resistance is a root cause for many problems, and we don't really understand what it is. Actually, we can measure it, but we don't understand the underlying molecular function, nor do we really understand how to treat it. Now, we were interested in this problem and re-examined a tissue that is linked to insulin resistance, which is the adipose depot. When you enter into obesity, as it was mentioned, there are many healthy obese, but most type 2 diabetics are drawn from the healthy obese group, and there is something that converts them to being insulin resistant, and we do not know what that mechanism, that conversion mechanism is. But we were studying the difference between lean and high-fat diet mice using a particular kind of screen. I won't tell you exactly how this was done because we don't have time. But the most dynamic gene that was changed in moving mice from a fasted state to a fed state to a high-fat diet state was FGF1, an old growth factor, the founding member of the FGF family. And that was really a bad result for the postdocs who were doing it because almost no one studies FGF1 function anymore because knockouts of FGF1 have no effect. And there's no known endogenous physiologic function for FGF1, and they said this result sucks, but it was the big gene that moved. So the question is, what had been missed, if anything? And we began to explore this by looking at FGF1 knockout mice. Now, FGF1 knockout mice, unfortunately, at the very top of the list, are the only FGF knockout that are normal and have no established physiologic role. And if you look at all the others where there are genetics, there's a lot to be said about them. It's FGF1 at this stage has become so boring that even the puppy thought it was boring. And that's pretty bad when a puppy thinks it's boring. Because everything is interesting to a puppy, but if it's not interesting to a puppy, it's not interesting to a postdoc. So we had a real issue, but we had to ask what was being missed for the more than 30 years that FGF1 had been on the table, and why has its function basically been stopped. And so we turned to FGF1 knockout mice. Now, Claudio Basilico, who created these about 15 years ago, sent them around the world. He had no function for those mice. The more than 20 labs that received them never had any function for those mice, and all the colonies completely disappeared except for one last colony that Claudio Basilico didn't know about. It was at the Salk that Steve Heinemann had, and he was going to discontinue the colony. He sent an ad out, does anyone want them? And so we were into this problem. I didn't know we could get them, but they're right at the Salk. So what we did that no one else had done is put them on a nutrient stress. Now, nutrient stress, in this case, is going to be a high-fat diet, but it can also be a starvation diet, and I'll show you both. when you take this strain with no known physiologic effect, suddenly, and I'm just going to say the knockout is the red line, and green is the wild-type mice that have FGF1. They both become glucose intolerant, but the one missing FGF1 becomes much more aggressively glucose intolerant, much higher glucose excursion. Insulin is less effective at lowering glucose, as you can see here, so there's much more insulin resistance if you are missing FGF1. So this gets us to why I was introducing that theme. If you look at what's happening in these knockout mice, you see that while normal mice get a fatty liver disease, if you're missing FGF1, you get a much more aggressive steatotic hepatitis or fatty liver disease. So we're having liver inflammation and steatosis. So now those were the first two phenotypes now for FGF1 in vivo. The third, which is striking, is a phenotype that probably no one in this auditorium has previously seen, and that is adipose tissue fibrosis. And adipose tissue fibrosis, the adipose depot is normal on chow, but if you put them on a high-fat diet, the entire depot becomes severely fibrotic with bridging fibrosis. And this is the fibrosis here, Mason trichome staining. And this kind of fibrosis around an adipocyte is really hitting the one function that is core to the adipocyte, which is the ability to expand and contract. Flexibility in most tissues is very important for dynamic blood flow and other functions, but in the adipocyte, especially where the cell can change its volume 3 to 10-fold, depending on diet and exercise and genetics, not being able to change size is devastating. So that's a fourth major phenotype. Now, I'm going to show you another phenotype that's even more striking, possibly, and that's if you take nutrients away. So the adipocyte has to both expand to store and then has to contract. It has to give it back the nutrients. But these adipocytes can't really do that. And the result is this incredibly exuberant induction of the pancreatic lipase system, the enzymes that dissolve fats that are not supposed to be expressed in the adipose depot, become highly expressed, and it starts dissolving the fats to release them into the body by producing the entire lipase system. Colipases, the lipase receptors, elastase, coelastase, to exuberant, it's hundreds or 500, 1,000-fold induction because the base levels for these are very low. The result of producing pancreatic enzymes in the adipose depot is that the depot itself undergoes a spontaneous necrosis. So you go from being dysmorphic to fibrotic to necrotic, and these pieces of formerly living adipose tissue now have broken off. So we open up these animals and you see pieces of the adipose depot because of this hydrolysis, lipolysis that's going on, and it's a remarkable phenotype. So these mice went from no phenotype, FGF1 doesn't do anything, to this crazy phenotype due to nutrient stress. So a key feature of evolution is the ability to manage nutrient stress. And if you do not have FGF1, we would not have evolved because it obviously is needed to manage the extremes. When there is food, you have to eat it. When there isn't food, you have to survive starvation. Those are the key filters for organisms and genomes, and that's a driving force for survival. So in this first part, this began by looking at how PPAR gamma manages nutrients in the adipose depot. We're looking for PPAR gamma target genes. FGF1 is a direct target gene, so nutrients come in like from high-fat diet or drugs such as the glitazones or TZDs that bind to this nuclear receptor. This is the environmental signal that acts on a nuclear sensor. The transduction of this process is FGF1. And in the adipose depot, it's required for adipose remodeling, expansion and contraction. If you have it, you're insulin sensitive. If you don't, you're insulin resistant. And so this was an exciting, now it became an interesting project. And we had a new model for FGF1, it's linked to PPAR gamma, and maybe it could help us unravel the mystery of how PPAR gamma is a target for an important class of drugs, the glitazones, but a troubled class of drugs as well. They're very potent insulin sensitizers when you activate this nuclear receptor, and they're the only class of drugs that have that particular insulin sensitizing property, but there are many other problems linked to the drugs. There's weight gain, there's edema, so there's increased adipogenesis, fluid retention, bone loss, cardiovascular problems, and so the drug is still widely prescribed, but it's not so popular in Europe, and it does have problems. So if this is important if FGF1 helps to control insulin sensitivity in a normal mouse, and the knockout is insulin resistant, could we explore this pathway in the setting of a high-fat diet where you have developed insulin resistance? What if we gave FGF1 back to this insulin-resistant diabetic mouse? And it's not a great experiment conceptually because FGF1 is one of the stickiest proteins. It's a heparin sulfate binding protein. It's produced widely in our body, but there is no serum level at all. If you measured your serum level at any time, you'll never see it because it sticks where it's made. We decided to do a process called, we named it endocrinization. It's not a real word, but now we're making that a word. And endocrinization is taking a protein that it normally doesn't circulate and simply introduce it into the body by a different route of delivery that involves a needle. If it involves a needle, we're calling it endocrinization in the sense that we've liberated it from its site of production. To get to a tissue, it has to come from the outside in, not from the inside out. I should also mention in humans, it has a half-life of 10 minutes or less. Same is true in rodents, so if you put it in serum, it's basically gone very quickly. So endocrinization can be mediated by IP injection, IV injection, sub-Q injection, okay? And the way we're gonna do it is inject a single dose of FGF1 into diabetic mice, type 2 mice, and see what happens to their glucose levels. And we got a remarkable effect. One single injection in the OB mice, a very difficult strain to treat, at 0.5 mg per kg of human FGF1, leads, this is a single injection within a few hours, to near normalization of glucose in a very difficult mouse model to control. And this is a single injection. And this can be achieved by, in this case, this was IP. It can be achieved by sub-Q. If you give it by IV, then the normalization is achieved within about 2 or 3 hours. So a single IV injection of FGF1 normalizes glucose, and there's nothing that is more potent or rapid in lowering glucose that I know of, other than insulin, than FGF1. And so it's really striking. Now, a lot of glucose-lowering agents tend to desensitize. Does this desensitize? Well, we've given it now chronically every other day for very long periods of time, and there's no desensitization. There's also no dose toxicity with this drug. You cannot get better than normal, so you cannot lower it beyond normal levels, and achieves that normal level within basically the first day and then stays there as long as we treat it. And that's for months now in these animals. And this is sub-Q now, injection, not IP. So it's really a phenomenal effect. Other components that are linked to this endocrine FGF1 is systemic inflation and for macrophages and neutrophils and monocytes. There you see a great lowering of inflammatory systemic inflammation. Again, with Jerry Olewski, Mark mentioned Jerry, who's the local expert. the glucose infusion rate, the amount of glucose that can be managed by the body, goes dramatically up, nearly doubles in these animals. They can handle almost twice as much glucose. The insulin-stimulated glucose disposal rate, how much sugar can be pushed into muscle by insulin, is dramatically elevated as well, almost double, about 80% increased. So these animals become insulin sensitized by injection of FGF1, and the insulin sensitization is equal to the TZDs. But oddly, TZDs take a long time to fully insulin sensitize patients. This happens in a way that we don't fully understand, but it happens very rapidly, and it's a long-acting effect. Now, therapeutically, FGF1 is not the happiest molecule to give therapeutically in the context of concerns about it's a growth factor. Although I have to say that when you look at the literature, it's a very poor growth factor, but it is a growth factor only in vitro. It's the only place where it's been demonstrated to stimulate growth is in fibroblasts in a dish. But because it's been around for so long and it was the first FGF isolated, the field had mutated basically every single amino acid in FGF1 and looked for its function on growth. And there are many, many, quote, growth dead or mitogenically inactive FGF1s. So we collected and made these. We synthesized the mitogenically dead FGF1s. Here's one of them. And this was a deletion of the amino terminus, and that just prevents growth. So we call that delta NT. This is one example of more than a dozen. If you look at its ability to lower glucose, and that's it right here, you can see that a mitogenically dead FGF1 is every bit as potent as the wild type. So structurally, you can separate growth and glucose lowering. So the metabolic effect and the growth effects, though in the same molecule, are not structurally linked. And that's important because now that opens up really a key safety window for considering this in a therapeutic setting. So we're very excited about exploring the ability to have a new class of insulin sensitizer. This has none of the side effects of classic TZDs. It does not cause edema, it does not promote weight gain, it has no effect on bone loss, and has no known cardiotropic effects. And so it may sound surprising, but oddly with this molecule, basically in a single day you can go from ultra high glucose to very normal glucose levels, and you can sustain that over a long period of time. Okay, so this is a brief summary of that part. FGF1 endocrinized, hits muscle, liver, adipose depots, lowers glucose output, increases insulin sensitization, lowers triglycerides and free fatty acids, improves macrophage polarization. And interestingly, we didn't show this, but Jerry Schulman, who was a reviewer of our paper actually, published shortly after our paper was published that injection of FGF1 into the brain was able to recapitulate many of the effects that we've been seeing, and speculates that the brain may be the target for what we're seeing. Now, we don't fully agree with that particular conclusion, although I don't dispute the result that he sees. Because if we knock out FGF receptor in the adipose depot, we lose the effect. And so at least the adipose depot is important, and the brain is probably very important. Okay, I'm going to move on. It is this, which is very important because this treats the root cause of insulin resistance without these problems. All right, the second part that I want to say, and I'll keep moving as quickly as I can, deals with the entry of food into our body. We're about to have lunch, and your lunch could be this or it could be this, but either way, food is going to be coming into your body. You're going to create that pressure gradient. And the first thing that happens when food comes in is you go back to the pyramid and you'll see that you're now here. And as soon as you eat, your body releases bile acids to help facilitate digestion of the food. And when bile acids are released into the intestine, you activate the bile acid receptor FXR. So here's the gallbladder here. This is the receptor that receives it. The receptor is expressed all throughout the entire intestinal system. It's also expressed at many other places in the body. High levels in the adrenal, high levels in the kidney, high levels in gonadal tissue, high levels in the liver, high levels in macrophages, actually. And so it is not like this is just only in the gut. So we wanted to look at the nature of the progression of the nutrient signal. And this just illustrates that bile acids, while they facilitate digestion, and many people study them as digestive molecules, we look at them in an entirely different light. We look at these as hormones. Think of it as a modified form of a steroid. Steroids are produced from cholesterol. Bile acids are produced from cholesterol. Bile acids, such as CDCA, are very similar to ecdysone. There's a worm hormone called dafachronic acid that looks just like a bile acid, and it controls nutrient homeostasis in worms, the Dower phase of worms. And so bile acids, think of it as being hormonal. They're produced in the liver, they're released in the intestine. They are detergents. They have important roles in their properties in controlling the microbiome. But I'm going to focus on its role in energy homeostasis. They can interact with two distinct types of receptors: bile acid receptor, FXR, transcription factor, or TGR5. This is a cell surface receptor, a GPCR, that binds lithocholic acid, which is a form of bile acid that is produced by the microbiome. So two bile acid receptors in the intestine, and also they're in other places as well. This is the structure of FXR when we crystallize the ligand binding domain, and we also made a ligand at Salk that activates FXR, which is shown here, and that molecule is called fexaramine. And it's a novel pharmacophore, not a type that had been built by anyone else, and we just made it as a tool compound to really be able to play with the system, because we had trouble getting the molecules that pharma was producing. So we made our own. It's very different, and it turns out to be an interesting molecule. I'm going to skip this and come back to it a little bit later. Here's fexaramine, which is here. Here is a regular bile acid, which is here. So the point of this is that fexaramine is not a bile acid. It's a chemical that binds to the bile acid receptor, and it tricks the receptor into thinking a bile acid is present. So if you give this molecule orally, it interacts with the intestinal FXR system, but importantly, this molecule is not bioavailable. It stays in the gut. You take it orally, it stays local in the gut, trundles until it is eliminated from the body. And so it activates potently FXR in the intestine, but not anywhere else systemically. So when you eat, you have a polarized response to food, which is FXR activation only in the intestine. And that signal from the intestine radiates out to the rest of the body. So our drug, in a way, mimics the initial process of eating. It gives the body a signal that food is there, whether or not food is there, whether you've eaten or not. You're sending a bile acid signal into the body through the intestine. And that turns out to have a very big impact. And I'm not going to dwell on this, only to tell you that fexaramine really does not get into the body, and it does not activate endogenous targets. When we look at its effects in high-fat diet fed mice, if you give it at a common point, first of all, this orally active drug prevents weight gain. And we have other forms that are now more potent that actually completely flatten this or actually lower this curve for weight loss. In addition, the molecule is shown in red. Remember, it does not get in the body, but it's improves glucose homeostasis in about 4 or so weeks after giving it to these mice on a daily basis. They eat the same amount of food, the same activity levels. The result of this reduction in weight gain is improved insulin sensitivity, not the kind that we see with FGF1. This is linked to a change in weight, and also reduction in inflammatory markers. The weight change that we're seeing here is a failure of nutrients to be deposited in the adipose depot, so there's a great decrease. This is stuff from the PC, changing it into a PC system, but in subcutaneous fat or visceral fat, you see big decreases in the amount of fat, but no change, for example, in heart weight. Some decrease in fatty acid steatosis in the liver. So it has a weight loss component. Now, where's the fat going? There's another unusual effect that is not seen with other FXR ligands that have been built, but we do find it here. And that is the induction of a thermogenic response and the browning of adipose tissue with the increased expression of oxidative enzymes. We see the induction of a nuclear receptor called ERR gamma. It's a major target of PGC1 alpha. Both of these are induced. PGC1 beta is induced. Uncoupling protein is induced. And you get a response that's browning of white adipose tissue. Increased thermogenesis like we're experiencing right now. You burn more energy as a result of taking this oral drug. And I'm not going to be able to tell you exactly why, but when you do that and reduce your adipose depot through a signal from the gut, and that's a very interesting feature, that you wind up having improvement again in these metabolic parameters such as insulin is down, cholesterol levels are down dramatically, HDL levels are up, leptin levels are down because the adipose depot is down, resistin is down, and triglycerides are not changed. Systemic FXR acting drugs will lower triglycerides, but our drug does not get to the liver, so it doesn't lower triglycerides. You get other major improvements in inflammation. There's TNF-alpha, for example, as one. You can look at other ones that you like. I like IL-17 because this is an important cytokine in the intestine. It helps to manage the microbiome through Th17 cells. MCP1 is also down. How does it work? It's going to be activating the genes that FXR likes to activate in the intestine, which are quite a few. A key hormonal trigger is FGF19. It's a gut hormone that is released upon eating. It goes right to the liver and manages bile acid homeostasis. So this is the secreted hormone in FGF as well that acts in the liver to control bile acid flux, which is important during and after a meal in both the prandial and postprandial state. It induces a series of other genes, as you can see here, and you can kind of go down the list in which we are doing and trying to dig into the nature of this gene expression profile that is triggered by FGF. By FXR, FGF19, it's 15 in the mouse and 19 in people. Now, two genes that get induced are interesting genes. One's called occludin, and occludin tightens the gap junctions in the gut, and when you eat, there's distension, there's physical distension, and there's a lot of endotoxin from the microbiome and actual bacteria that get into the system. So when you eat a meal, you have bacteria now that are circulating around in your blood. We find that high-fat diet mice that are chronically fed high-fat diets and weight gain, have a fair and measurable living microbiome inside the adipose depot. We isolate live bacteria from the adipose depot. And so occludin prevents the translocation of bacteria from the gut into the circulation, and that's important. Mucin is this gelatinous type of substance that coats the gut and protects it again from the microbiome, and these are two sort of factors that promote gut health. All right, this is, and I should say it also suppresses the microbiome itself. This is the number of bacteria that are measured at the cecum, and just giving this oral drug suppresses the microbiome. If you look at the gene ontology, it reads it out as antimicrobial, and that's a very interesting feature because high-fat diets tend to cause microbiome blooms, and this takes you in the opposite direction. Okay, I'm just going to skip through a few of these. The liver is improved, and you can look at some of the parameters that from steatosis to ALT levels that happen when you take fexaramine, and we're still trying to work our way through what really happens. Now, an important feature that happens is that the body thinks that you've taken a big slug of bile acid because it's hitting the bile acid receptor with a very potent signal. Now, if the body registers a lot of bile acid, it lowers the bile acid pool because it doesn't need to make that. So when the bile acid pool is low, you convert cholesterol into bile acids, and as a result, a low bile acid pool leads to low cholesterol. When cholesterol is low, you actually get high HDL. And so this shift in the bile acid pool is actually very important because it lowers cholesterol approximately this amount as well and increases HDL. It also changes the components of this pool, and it induces LCA, lithocholic acid, and taurocholic acid is reduced. Now, this is interesting because LCA is the best bile acid, secondary bile acid ligand for TGR5. TGR5 is the other receptor that I mentioned. It's in the gut and it's in other tissues in the body. This change in bile acid pool is not just restricted to the gut. This happens body-wide as well because there's a lot of bile acids that are circulating in your blood. And that activates this thermogenic response, at least in part, we believe. It's not the whole thing, but TGR5 downstream of FXR, downstream of the bile acid pool change turns out to be an important feature. So the summary of this is that second part, another environmental signal, which can be your diet, triggering bile acid release, or a drug that is not a bile acid but that triggers the same target. Here's your sensor again. The transducer is going to will be gut peptides such as FGF19, but probably many other gut peptides that we feel are important in this. Also, the microbiome is improved dramatically. Gut inflammation is also a major beneficiary of this process. And all of this is a drug that does not get into circulation, leads to all these improvements in the liver, cholesterol, glucose, bile acid, and biliary cholestasis or bile flux, and this impact on white adipose tissue. So we're excited by this pathway for a different reason, but it's really when you trace how the body manages food and try to recapitulate that through sensors that often are related to the actual diet itself, you can trick the body into thinking that you've had a good meal, actually, but without any calories. And the body responds, doesn't know that there were no calories and that you didn't actually eat something. And you wind up losing weight in that process. It has by virtue of the fact that it does not get in the body, there's a certain safety benefit to a drug that doesn't get in. And we're interested and we like that feature. It mediates this postprandial response to bile acids and to eating without having to actually eat. that gives you many metabolic effects. In particular, it gives an effect that's very similar to bariatric surgery. So bariatric surgery, we do not really know how it works, but very shortly after bariatric surgery or vascular sleeve gastrectomy, the body begins to normalize glucose and insulin, and there's weight loss. It has been described by Randy Seeley and others that that process now requires FXR. It's lost in an FXR knockout mouse model. And so maybe FXR is a key mediator of this surgical procedure. The gut restriction is a great feature, and these are some of the targets. That's the Randy Seeley paper that I had just mentioned about vertical sleeve gastrectomy. I'm just going to end, this is one of the last pictures that we had of Wiley. And of course, it was in Hawaii. That's where he passed away. This is the day before. And of course, where does he have his Wiley picture taken? Danger, steep cliff. And Wiley loved this, and it's it was a great loss for all of us at Salk, for me as a personal friend. But he had landed a big impact during life, and we celebrate his science and inspired by it. The FGF1 project was done by these three postdocs. Jay Su now has his own job. Hans Jonker has his own job. Miriam Ahmadian, that's hers. And I'm going to skip over to FXR. Sung Soon Fang also has his own job in Korea, along with Jay, same guy, and Liz Yu. And this group right here were in both projects, and they really helped to guide with valuable information and input. It's a senior group of folks in my lab, and want to acknowledge them for that. And then a number of great collaborators on these two projects that have really helped to push this through. Really, it takes a village for this kind of stuff. So again, I really appreciate the opportunity to come to Siena, and also to honor Wiley and to be able to give you guys this talk. So thank you very much.